Good day, sir. I am Gracechel Paypon, and today I will be reporting the research we had done entitled AIDS Pandemic Understanding It More. So, the virus known as human immunodeficiency virus undeniably affect millions of people worldwide, having a significant global impact. The so called human immunodeficiency virus, or HIV, is the one responsible for causing acquired immune deficiency syndrome, or AIDS. So, in the US, the recognition of the acquired immune deficiency syndrome started about the year 1981 as a result of the increasing number of patients residing in San Francisco or New York, may it be male, gay, and adult sex, which demonstrated immune suppression of the immune system, pneumonia caused by pneumocystis carini and Kaposi sarcoma. So HIV is the virus that causes AIDS. This infection is spread by viral particles that are typically present in blood, sperm, and the vaginal fluid of an infected person to an uninfected person, and the main method of prevention is the use of condom. So, vertical transmission is the type of contagion that occurs when the mother's virus is passed on to her child during pregnancy, giving birth, or even breastfeeding. So, following the acquisition of the viral envelope glycoproteins, when the virus enters the defense cells, it causes the immunological alterations with direct lysis of the primary source of the lymphopenia, which is the functional impairments caused by CD4 T cells or the cluster of diffraction, particularly group number 4 immunological response. So, antiretroviral medication is used for this treatment. It is the medicines that try to stop the HIV replication cycle. So, despite the reason that HIV virus is widely known, the increase in number of infected individuals is still a global concern. Through this conducted research, it desired to show how HIV virus develops to AIDS, its point of entry, mode of reproduction, symptoms, data on trends, as well as prevention and medication contributing to the increase in awareness and result in the decrease of the number of cases. So, in conducting this research, the materials and the method we use is to carry out a keyword analysis in the database searches such as the Google Scholar and the Google. We choose the papers that were most pertinent to the years 2000 up to the present. So, the origins of the AIDS pandemic. So, HIV-1 changes around 1 million times more quickly than DNA of mammals as a result of the HIV-1 reverse transcriptase proneness to errors and the rapidity of the viral production. This inclination toward quick genetic change has given a rare chance to obtain understanding research on the AIDS pandemic, onset and spread its genesis statistical and phylogenetic analysis dated the HIV-1's last common ancestor group M through roughly 1910 to 1930 with limited confidence intervals. So these suggest that following the initial appearance of the pandemic HIV in colonial West, it expanded throughout the Central Africa for around 50 to 70 years before anyone noticed it. Additionally, the location of the early outbreak has been determined. So, according to molecular epidemiological research, the majority, if not all, of the early diversification of the HIV-1 group M most likely to took place in the vicinity of Kinshasa, which was formerly known as Leopoldville. So there, other lineages that have stayed localized to this region as well as all known as HIV-1 group M subtypes were discovered. So the early HIV-1 group M strains were also found in Leopoldville. So examination of the genetics of contaminated blood and tissues gathered from citizens of Kinshasa in the years 1959 and 1960 demonstrated that the HIV-1 strain had already changed by that point into many subtypes. So finally, according to the demographic statistic, the pandemic HIV-1 outbreak occurred while West Central African cities' urban population were growing. The biggest city in the area at the time, Leopoldville, was a likely target for a newly emergent virus. Additionally, which were important trade and transport routes at that period, would have connected the chimpanzee reservoir of HIV-1 Group M at Leopoldville on the banks of the Congo and Southeast Cameroon rather. So therefore, Leopoldville or the Kinshasa is believed to be the origin of the AIDS pandemic based on all the available data. So the global spread of HIV-1 group M involved a number of populations bottlenecks or founder events which caused distinct group M lineages, now referred to as subtypes, to predominated in the different geographical areas. A, D, F, H, J, and K are the nine subtypes of the HIV-1 group M that are now recognized, and there are more than 40 distinct circulating recombinant forms that were produced when numerous subtypes infected the same population. Some of these subtypes or CRF's migration routes have been able to be tracked. Subtype C was and introduced to and predominates in southern Africa from where it moved to India and other Asian countries. In contrast, the subtypes A and D originated in Central Africa but ultimately formed epidemics in the Eastern Africa. The majority of HIV-1 infections in Europe and the Americas are the subtype B, which developed from a single African strain that is thought to have initially moved to Haiti in the year 1960s before moving on to the US and other Western nations. So, although the recombination event that gave rise to the CRF01 most likely had place in Central Africa, this viral lineage was first identified in the late 1980s when it propagated among intravenous drug users in Thailand and caused a heterosexual epidemic. So, the Southeast Asia's AIDS pandemic is now dominated by CRF01. So, recent investigations have revealed that viruses of the distinct subtypes differ in their epidemiology, even though the initial spread of these subtypes or CRFs may have been primarily influenced by chance events. Subtype B, for instance, has been linked to the increased pathogenicity as well as the rise in the prevalence of the cognitive decline and AIDS dementia. Therefore, it would seem that both the genetic and the biological diversity of the HIV-1 group M subtypes and CRF are growing. After knowing the origin of the AIDS pandemic, we have here the modes of transmission of the so-called virus. So most of the reported cases of acquiring HIV virus are through a direct anal and vaginal sex, sharing contaminated and unsterilized needles, syringes, and other drug injection equipment. Some cases are because of mother who has HIV and they can transmit it to their child either before or after childbirth and or through breastfeeding. So first we have here the sexual, which is through direct transmission through sexual intercourse or unprotected sex, either vaginal or anal. So for the anal sex, it is the riskiest type for either getting or transmitting HIV. Factors affecting the easier transmission of HIV through this type is first being the receptive partner or you are on the bottom part. It is riskier than being on top for the reason that the rectum's lining is thin and may allow the HIV to enter the body during the anal sex. So while being the top also has its risky. If you are on top, HIV can enter the body through the opening at the tip of the penis or the urethra, the foreskin if the penis isn't circumcised, or small cuts, scratches, or open sores anywhere on the penis. And for the vaginal sex, compared to the receptive anal sex, the vaginal sex is less risky. However, remember that still both individuals have a high risk of acquiring HIV infection. So during this type of sex, the HIV will enter the body through the delicate tissue which lines both the cervix and the vagina. So the vaginal fluids as well as the blood carry the HIV which can then be passed through the urethra or the opening at the tip of the penis and then the foreskin if the penis is not amputate or if there is any presence of even small cuts, scratches, or open sores that can be found on the penis. And then for the needle, or the instrument related sharing blood contaminated needles or exposure to contaminated instruments as well as needles that is unsterilized or used without being sterilized can then um, transmit HIV. So used needles, syringes, and other injection equipment might still have contained blood that carries the HIV infection. And aside from HIV infection, the risk of getting hepatitis B and C and other diseases is also high due to the contaminated injection equipment. And then after that is we have the another transmission which is the maternal or the childbirth or breastfeeding. So although nowadays this type of transmission is less common because of the advancement in both the HIV prevention and treatment. Still, there is a possibility that HIV can be transmitted by the mother to its child during pregnancy, birth, or breastfeeding. So, this type of transmission is also known as the perinatal transmission or the mother to child transmission. So, during the pregnancy, the HIV infection can be passed by the mother to her baby through the placenta. And then during the childbirth, the exposure of the baby to body fluids from its mother is high, and during this time, the mother's amniotic sac breaks and other body fluids such as the blood is also present during this time. And once this happens, the risk of HIV transmission is higher. And during breastfeeding, HIV can be transmitted given that this fluid carries the HIV and can be transmitted as cells associated virus or cell free virus. The amount of the virus as well as the viral load that are present in the breast milk are the principal factors that affects the rate of the transmission. And then lastly for the modes of transmission is we have the transfusion or the transplant related. So through exposure to body fluids such as the blood products, semen, and the vaginal fluids, whether you are directly or when you are directly in contact with the body fluids such as the blood, semen, and the vaginal fluids, then you are at risk of getting HIV as these body fluids carry the HIV infection. Following the modes of transmission is we have the mode of the reproduction of the HIV virus or the disease stage of it. So first is we have the acute HIV infection. So it is the earliest stage of the HIV infection that generally occurs within 2 to 4 weeks following the actual exposure and infection of the virus. So in this stage, the body mounts a defense against an outside threat after recognizing it. So the HIV multiplies quickly during the acute stage of infection and spreads all over the body. So the immune system's infection fighting CD4 cells are attacked and destroyed by the virus. And the risk of HIV transmission significantly rises during the acute HIV infection stage due to the extremely high HIV blood levels. It's critical to take into action the possibility that you may have HIV at this stage and start the process of asking your doctor about testing. So the incubation period for the acute HIV infection starts within 2 to 4 weeks after the exposure to the virus and might last for up to 2 weeks, just like the typical flu. And the symptoms are mainly flu-like symptoms, which can include headache, diarrhea, nausea, vomiting, aching all over the body, being very tired, having a rush all over the body, and a sore throat and a high fever. And all of these symptoms are the result of your immune system fighting against the virus. Then after the acute HIV infection stage, we have the chronic HIV infection. This is also known as the asymptomatic HIV infection or the clinical latency, which is the second stage of the HIV infection. So once the immune system has the virus under control, the illness enters the chronic stage. HIV will hide during the stage where it will exist in different cells and tissues all across the body in a dormant state known as latency. So HIV continues to slowly and invisibly replicate in the body even while in latency, depleting the immune cells called the CD4 T cells, and it's possible for people with persistent HIV infection to be symptom free. However, there are several general symptoms that could manifest when immune insufficiency worsens, and this includes oral candidiasis, unexplained fevers, and severe or uncontrolled diarrhea. So the incubation period during the chronic HIV infection might go up for 10 years or decades without another sign of HIV. So always remember that during this HIV chronic period, it is very easy to transmit the virus to someone else. And the symptoms during this time includes during this time, there are not typically symptoms for a while. So after the first symptom to show up, usually years later, includes fever, feeling tired all the time, swollen lymph nodes, and weight loss. You might also notice diarrhea, oral yeast infections, and recurring outbreaks of herpes zoster or shingles. And several general symptoms that could manifest during these stages include a fungal infection known as the oral candidiasis or thrush, results in the development of the creamy white lesions on the tongue sides and mouth lining. Also, we have undiagnosed fevers and night sweats so intense that they soak through your bed linens and pajamas. And lastly, we have the severe diarrhea that is uncontrolled and lasts for more than 3 days. Then after the chronic HIV infection, the virus will advance to AIDS infection. So it is the final and the most dreadful stage of the HIV infection. So the immune system has been badly harmed by the HIV during this time, making it possible for the body to resist opportunistic infections. So people with HIV are diagnosed with AIDS based on the CD4 cells in the body. If the CD4 count less than 200 cells per cubic milliliter of blood, then the normal count of the CD4, which is 500 to 1,200 cells per cubic millimeter, or if they have specific opportunistic infections, then the patient is considered to acquired AIDS. So after receiving an AIDS diagnosis, a person may have a high viral load and be particularly likely to infect others with HIV. People with AIDS typically live for 3 years without treatment. So symptoms for this stage have worsened. So viral, bacterial, fungal, parasitic, and malignancies like non-Hodgkin lymphoma and invasive cervical carcinoma rather are among the symptoms of the advanced HIV and AIDS infections. And organs and other parts of the body are affected by these infections that includes our lungs due to bacterial pneumonia, tuberculosis, pneumocystis, and pneumonia, our skin due to shingles or the Kaposa sarcoma for the digestive system, the Mycobacterium avium complex. For the brain, we have AIDS dementia, and then for the eyes, we have herpes zoster ophthalmicus, and for the blood, we have Salmonella septicemia. So as you can see here, we have the figure one, which is the HIV progression. So the first picture, as you can see, that is the healthy CD4 cells before the HIV or before the body is infected with HIV infection. And once infection occurs, as you can see, um, our body, which is the acute HIV infection, our body, I mean the virus, HIV virus multiplies quickly during the acute stage of the infections and spreads all over the body. So that is the red dots over there. And then later on, it will advance to chronic HIV infection, which is our immune system has the virus under control. However, the HIV continues to slowly and invisibly replicate in the body, even while in latency depleting the immune cells called the CD4 T cells. And then later on, if it is left untreated, then it will advance to AIDS infection, which is the final and the most dreadful stage of the HIV infection. Just during this time, the immune system has been badly harmed by the virus, making it impossible for the body to resist the opportunistic infection. So much for that. We have here the pathophysiology of AIDS. So HIV utilizes the CD4 cells, molecules, and the chemokine receptors to bind and to enter the host T cells. So several HIV encoded enzymes and HIV RNA are released into the host cell after the attachment. And then the reverse transcriptase rather, or an RNA dependent DNA polymerase is required for the viral replication in order to copy the HIV RNA and create a proviral DNA. So this copying mechanism is error prone, leading to frequent mutations and new HIV genotypes. So these mutations make it easier for the HIV to develop that can withstand immune system and antiretroviral treatment control in the host. So integrase, another HIV enzyme, is required for the integration of the proviral DNA into the host DNA once it has entered the nucleus of the host cell. So the integrated proviral DNA is duplicated along with the host of the DNA during every cell division. And the HIV proviral DNA can then be translated into the HIV RNA and the HIV proteins like the envelope glycoproteins 41 and 120. So these HIV proteins are put together, form HIV virons at the inner membrane of the host cell and bud from the cell surface inside of a modified human cell membrane envelope. numerous variants could be produced of each host cell. Then, after the budding, the HIV enzyme protease breaks down the viral proteins, maturing the immune virion into an infectious mature virion. Then, greater than 97% of the plasma HIV variants are produced by the so called infected CD4 cells. HIV can be reactive in a fraction of the infected CD4 cells. Like, for example, if the antiviral therapy is discontinued. And during the mild to severe HIV infection, 108 to 109 variants are produced and deleted each day. The HIV virus has an average half life of 36 hours in plasma, 24 hours inside the cells, and 6 hours outside the cells. So, roughly 30% of the entire HIV burden carried by an infected person is turned over each day. Additionally, between 5 and 7% of the CD4 cells can change every day, and the full CD4 cell population does so every 2 days. So, consequently, AIDS develops from HIV replicating continuously and consistently, which causes the virus and the immune system to kill the CD4 cells. So, additionally, the high rate of transcription mistakes made by the HIV reverse transcriptase and the volume of the HIV replication led to many mutations, increasing the likelihood of creating strains resistant to the host immunity and drugs. So, as you can see, here is the pathophysiology of the AIDS. So, we have here the factors, which is the HIV variant that becomes matured. Then, after the HIV variant being matured, the HIV then fuses with the CD4 cell receptor, and the reverse transcriptase changes the RNA into the DNA. And there is a nucleus entry, which is the integration of the provirus into the host cell genome, resulting to the HIV DNA provirus. And then HIV RNA transcription, which is the release in nucleus or the synthesis of the HIV proteins assembly of the virus. Then the migration to cell membrane happens. Then a budding a new HIV um, becomes becomes the result, and then it produces a new HIV variant, and then transmission occurs. Then after that is we have the prevention for this HIV virus infection. So the Department of STD, AIDS, and Viral Hepatitis thoughts that using condoms during all sexual interactions reduces the risk of HIV to 5%. So contact with the unprotected individual should be also avoided when it comes to blood and blood products, semen, body fluids, and breast milk from infected individuals. And the prenatal testing should be done on pregnant women to look for the viruses, and if tests come back positive for HIV, treatment must be also started right away to protect the embryo from the contamination. So, vertical transmission or passing the virus from the mother to child can then happen frequently during pregnancy or delivery, and there is also a chance that it can happen through breast milk. So, health personnel may contract HIV by occupational transmission, which results from an occupational mistake where they may be hurt by cutting or sharp objects contaminated with the HIV positive patient blood. So the extent and the deep of the cut, as well as the presence of the blood in the accident causing tool and the patient's status as a source with a high viral load and advanced immunodeficiency signals are some characteristics that may lead to occupational contamination. The effective application of the biosecurity standards in an invasive procedures to implement new technologies, as well as the study to determine the risk factors associated seeking their elimination, are among the control measures that should be adopted in order to minimize the risk of the transmission by this type of contamination. Then, irregular testing for HIV in blood donors has drastically decreased this method of transmission of the virus. And effective HIV prevention is crucial, remember, for controlling the epidemic, and thus as our public health initiatives to reduce the use of contaminated needles by the intravenous drug users and common use, even among HIV positive partners, prevent the reinfection of viral strains that are drug resistant and can cause serious health complications, and raise public awareness of HIV AIDS. And then we have the treatment for this infection. So, all medications required for the treatment of the patients with HIV/AIDS are currently used in Brazil. For classes of the antiretrovirals, which includes nucleoside reverse transcriptase inhibitors, nucleoside inhibitors, no reverse transcriptase inhibitors, protease, and the integrase inhibitors are thought to be more effective and less toxic. Brazil was the first nation to make these drugs available free of charge in the year 1996 through the Unified Health Reverse Transcriptase. It is an enzyme that is inhibited by the antiretrovirals or the RTI. So during the first phosphorylation, nucleoside inhibitors of the metabolites became active. So due to the challenges in creating the monophosphorylated molecules, similar medications with the nucleotide phosphate grouped in their structure, which only need two phosphorylation to prevent RNA from being converted into DNA, have been developed. However, the NNRTI antiretroviral class is thought to bind to an enzyme's allosteric location non-competitively. This contact limits the flexibility as well as the mobility of the active site, which is crucial for for the creation of the DNA double helix, which significantly lowers the effectiveness of the enzymes. And by interfering with the final stage of the viral replication, protease inhibitors stop the development of the new virus. The polyproteins GAG and the GAG pol, which are responsible for the creation of structures and the functional proteins, formed viral particles that are produced by the enzyme protease. Last but not the least, according to Bacelar, there are known medications that inhibit integrase, which are regarded as one of the emerging types of the anti-HIV drugs. And the proviral DNA is inserted into the host chromosome by the integrase enzyme, which also catalyzes the incorporation of this proviral DNA into the effective cell's genome, which is necessary for the viral replications. And then lastly, for the conclusion, so, to summarize everything, acquired immune deficiency syndrome, also known as AIDS, is caused by human immunodeficiency virus or AIDS. So, the infection AIDS caused by the HIV is spread by viral particles rather that are typically present in blood, sperm, and the vaginal fluid of an infected person to an uninfected person. So most of the reported cases of acquiring the HIV virus are through direct anal and the vaginal sex, sharing contaminated and sterilized needles, syringes, and other drug injection equipment, as well as during or through childbirth and breastfeeding. This begins its early stage, which is the acute HIV infection, where the immune system infection fighting CD4 cells are attacked and destroyed by the virus. Then later on, the HIV continues to slowly and invisibly replicate in the body, even while in latency depleting the immune cells called the CD4 cells during the chronic stage. And if virus is left untreated, it will eventually advance its stage, wherein the immune system has been badly harmed by the HIV, making it impossible for the body to resist opportunistic infections and reach its final and most dreadful stage, which is the AIDS stage. So the so-called antiretroviral medication is used for the treatment, which is the medicines that tries to stop the HIV replication cycle. So that would be all for my report about the AIDS pandemic, understanding it more. Thank you.